So today I'm going to do three things. I'm going to try to explain you a bit which kind of questions we try to address in our lab. Okay. Well, I only talk about one of the questions, but then I also talk about how we address this question, and then I will show some results. Okay, not very conclusive, but hopefully they will address some of the questions. Okay, so imagine you could build any gene network in development and check out which kind of things they can produce. The kind of question is like, how should a network be in order to build a complex and robust body? Okay, for example, morphology. And by morphology, I mean the distribution of cells in space and the extracellular matrix. Okay. So, and of course, this has to be in development because morphology doesn't come to be like it is just by chance or by genes or something like that. But it's a process through which over time we go from one cell to different cell types, and especially those cells have a specific distribution in space. So we generate some spatial information, and we call that development. Okay. So the question is like, is there something in common among the developmental networks who can actually lead to complex robust morphologies? For, I don't know, perhaps something about the gene network topology, perhaps something about the gene cell behaviors involved or how they get coordinated. Can we acquire some general principles about how should be gene networks be constructed? Or more specifically, how did evolution did it? I mean, because I'm not an engineer, I just don't want to understand how to do it. I want to understand how it has been done. Okay. So for complexity is like about how complexity arises. Okay. For robustness is about how can development be stable in spite of the noise intrinsic to the molecular and cellular level. Okay. So as you know, molecules move by Brownian motion, and cells suffer all kinds of stochastic processes. So they are not really stable. They move a bit. Okay. In spite of that, we think that development is stable. And there's two classical views about that. The first one is by Waddington and colleagues, is that there is some specific mechanism of buffering that have evolved to render development stable. Okay, so it's some robustness of development with some property that has evolved on purpose somehow. The other way to think is that no, it's not. There's no really buffering mechanism, but there's just development somehow by its normal way of proceeding filters noise. Okay. And that's a classical in simplifying the two extremes about how people understand robustness in development. However, there's a kind of thing or kind of argument you should consider in relationship to those two kinds of arguments. So essentially, we can only say or claim the existence of buffering mechanisms if we have some expectation of how much noise at the morphological level should there be from the noise we know at the cell and molecular level. Okay, if we have such expectation, then we can say that there is a buffering mechanism or not, based on the fact of observing that there is more morphological noise than they should. That there is less morphological noise than what should expect. Okay, so in a way, if we don't know the normal way of proceeding of development, we cannot claim there is some buffering mechanism. In a way, we should somehow have an expectation of how robust should morphology be if those buffering mechanisms don't exist. Question is, essentially, we have this cell noise, a molecular noise, okay? Oops. Sorry about that. So we have cell noise and molecular noise. They affect development. By affecting development, they might affect variation, morphology. So essentially some morphological noise or some variation in morphology. The question is that in order to understand that, we have to have some theory about how development works, at least in some general properties, okay? And then maybe we found out that some cell processes or some gene network topologies, something else, have an effect on how this noise leads to this kind of noise over here, okay? Okay, so how can we address this question? So there's two ways. We can do it experimentally, or we can do it theoretically. Experimentally, we could do some synthesis biology and construct all kinds of gene networks and try to see what they produce in development. But that will be quite costly and tricky. Okay. We can also have an indirect approach where we try to study as many examples as possible of development. Okay, and that's what we all do to some extent. We study tooth development or leaf development or wherever. And from that, we try to extract some general principles about how phenotypes are constructed. Essentially, nobody is interested in wing development by itself or in tooth development. No. Okay, some people might. But in general, you use that as a proxy to understand how this morphological complexity and structure arises and how it comes that it's somehow robust, at least to some extent. So we can do that. We also do that on our lab, but we can also have a direct theoretical approach that is the following. We can build a general model of animal development, then construct a huge number of gene networks and try to see from the model what they produce, which kind of morphologies they produce, and whether they are robust or not. Once we do that, we can check which morphological noise we can expect from cell level noise in this model. So this will provide us some expectation of, okay, how much morphological noise should we expect in morphology? Is that the noise consistent with the one we observe in nature? If not, then there is maybe some buffering mechanism we're missing, okay? And also by checking which networks can lead to complex and robust morphologies, then we can see what they have in common and maybe hopefully extract some principles about how to construct complex and robust morphologies. So that's what I'm going to tell you, how we do that and what we learned so far. Why do we think we can do such a model? Okay, that's the first thing we might ask. So you may think, oh, these theoretical guys, they are crazy, they can think they can do everything. Okay, so there is a trick. The trick is that in development, well, development is very complex. There's all kinds of funny genes with funny phenotypes who are quite complex to understand and so on. But at the end, everything that happens in development happens because of cells, and cells can do a limited number of things. They can divide, they can move in an active or passive way, they can bind to each other and to the substrate, and as a result, they can change of shape. They can also contract, who might also change their shape, but they can also have effects over the whole morphology of the embryo. They can die, they can change of state, you can say they can differentiate, who essentially changes which of those things they do. Can change the shape, which is expressed, who affects what they do. And of course, they can secrete molecules who might affect what other cells do, or might actually be extracellular matrix who mechanically interacts with the rest of the body, okay? So in a simplistic way, you could see that development is just the coordination in space and time of those things that cells can do. Of course, for this coordination, you also need molecules or gene products if you want, and these also can do a limited number of things. And the results, the overall results at the high level, they are pretty complex, but at the low level, they only do three things. They bind to each other in a way that can be specific or not so much, it depends. They can change of conformation, like physically or chemically. For example, they can catalyze reactions by binding to the transition state of a reaction, or they can move in an active or passive way, okay? So the trick of that is that if you can implement to some extent all those things that cells can do and all those things that molecules can do, then you have, at least as a proxy, a model of development. And that's why we actually have. This is the model, okay? It's called Embryo Maker, the model. So we have two types of cells. We have cells who are actually cylinders. You should think about them as if they would be actual physical cylinders. And then we have cells made of spheres. Depending on the resolution you want, you can make that each cylinder is a cell, or that several cylinders are a cell, okay? Then they interact in a very simple way. If they get too close to each other, they repulse each other. And if they are too close to each other, but not too much, they actually bind to each other. And this addition can depend on molecules they express, okay? And we have some special rules for the cylinders who are epithelial cells. So they kind of try to avoid they avoid the epithelia to bend to some extent, or there's a force for that. Then within cells, we have molecules, and we simplify them as variables who represent the expression of each molecule. So they can actually interact with each other. So we have a gene network, and some of them can diffuse either within the cell or between the cells, and then actually act as a growth factor, so paracrine factors, if you want. Okay. And then cells can do the kind of things that they normally do. They can actually divide in a symmetric or asymmetric way. They can actually grow in a polarized or non-polarized way. Cells can polarize depending on the gradient of extracellular signals or molecules. They can die. They can secrete extracellular matrix. They can do the kind of stuff that cells are supposed to do in animals. Sorry, well, okay, so then we check that we can do all the kind of stuff they normally do, but I will skip that. I will show you that the model can do nice things. Could you switch off the lights over here, please? Because there's plenty of cool movies. Where? I mean, the white. So it's just too many stuff here. Oh, sorry. Okay, cool. So what we have here is just an example of the kind of stuff we can do with Embryo Maker once. Okay, now this has crashed for some reason. Okay, now. So what we have here is we give to the model some gene network, which essentially a user specifies a gene network, so a set of genes, which genes interact with each other, and which genes regulate some specific cell behaviors or mechanical properties in the cells where they are expressed. And we also provide some initial conditions. Here it's just some kind of blastula-like sphere of epithelium where cells express nothing except for one cell here in the middle who expresses transcriptional factor 1. And then the model predicts from this gene network and this initial condition, it predicts how morphology and gene expression should change over time based on this gene network. Okay. Cool. So we have some kind of general model of development. This is something you can download and use on your own will. We have used it to simulate several things. Then our aim, however, is to have this general perception of how networks should be to produce complex morphologies. And for that, we have this ensemble approach. It's very crazy, but it's very informative. What we do is we just wire genes at random and wire some of those genes to cell behaviors or mechanical properties. Okay, so here we have an example. This is a random gene network in which we have those genes. Some of them actually fuse between cells, some of them don't. And then this gene affects addition between cells, and this gene affects growth. Okay, and we start from this initial condition, which is like a blastula-like, where we have two genes, one in the anterior part and one in the dorsal part. Okay, so we actually simulate this gene network, we get this morphology. Cool, kind of a random larva-like morphology. And here we have another random network, we get this morphology. As you see, the initial condition has bilateral symmetry, and the result might or might not have it. Okay, so this morphology is relatively complex, but it's robust. This morphology is more complex, but it's less robust. Okay, so we want to have both. And just try to make a point here. We are not saying that gene networks are random, of course, but it's just a way for us to explore in an unbiased way the space of possible networks. Okay. So just the dynamics of the model are pretty simple. They are just like in animal development in general. You have some initial conditions, so that's a territory, a group of cells expressing the same gene. This gene promotes the secretion of a signal who actually reaches some distance and induces a new territory. So a different distribution of cells expressing the same gene. And this gene might actually activate other signals, and it might also affect, for example, cell growth or contraction. So there's a change in morphology. So if you if you change these kind of things over time in a gene network, what you get is the morphology changes. And that's what we're doing. We're not doing anything especially fancy at that level. We have a way to measure complexity. I won't go into that much, but just so you know, and any way we measure complexity, we get the same results. And we also have a way to measure robustness. We essentially compare how similar is the left side to the right side of the embryo, because we start from initially bilaterally symmetric morphologies. Okay. So what do we get in the sample? So we get a lot of different morphologies, as you could imagine. I'm just giving you a sample of here. The color is just to see the morphology a bit better. I'm not plotting gene expression. Okay. So we get plenty of them. So now we want to understand, okay, so in this situation, most of them are, of course, not very complex. Complex are rare, and the complex and robust are really rare. Okay. In a way, making complex morphologies, however, is kind of easy. If you just activate some cell behavior homogeneously over space, for example, contraction, you will get a very complex morphology. You get a morphology made of all kinds of invaginations positioned in random positions. Okay. So they are complex, but not very stable. Making a stable morphologies is also quite simple. You just do nothing, and that's going to be very stable. Okay, so you'll keep the same sphere, and nothing will happen. The trick is to get complex morphologies who are also stable. That's the trick. That's how the kind of thing that's difficult in evolution and is difficult in development. Also, we found out if we plot each of the morphologies in the ensemble, each of these random network morphologies, we find that the ones who are complex also happen to be less robust, was somehow, and not surprising, but not very nice. Okay, so now what we can do, and what we did, and what we are actually doing at the moment, is to follow in the model the morphology, the development of each morphology. because in the model we are God. We can actually see everything. We can have all the lineages. We can make all the knockouts you want. We can do everything. Of course, it's just fantasy, but we control it very well, so we can understand it very well. Okay, so we actually follow the development of many different gene networks, and we found that there's 6 main factors who actually explain how you actually get morphological noise from noise in the positioning of cells, so random movement of cells. Okay, just a bit. We have growth. If you see here, we have how much. So fluctuating symmetry. This is actually how different is the left side of the embryo from the right side of the embryo. So it's just robustness against noise. Okay. So and here's actually a number of cells, and each line is the development of in one gene network. Okay. As you see, it increases over time. Just growth by itself decreases robustness. Here you can see an example morphology. It starts being a bit, has these 2 bumps, plumps, or whatever bumps you call him, but then they start to get a bit different in each side and so on. Okay. We know why that happens. The explanation, it's not so easy to explain, but essentially what you have is that the epithelium grows, okay, and then there's this force against bending, I told you, who makes that if a cell reorients in space, it changes its direction of the apical basal direction, vector, then the neighbors will try to avoid that. So essentially all cells want to be aligned in the same direction, at least in the apical basal axis. But this force is local. You actually affect your neighbors. You don't affect very far away neighbors. So as the embryo grows, there is actually the different parts of the epithelium start to be uncorrelated in their directions. And this actually implies there's going to be formation of invaginations and evaginations. And we are pretty confident that happens in reality too. In fact, there is some evidence for that. Okay. One thing we can do to solve that is to make that this force against bending becomes more strong, and that's what we have here. So this best value is how strong is the force that epithelium make against bending, and as you increase it, you actually get less noisy morphologies. Of course, there are some more simple, so it's not so cool. In this case, a bit different. Here we have a morphology where there's this blue area who actually suffers apical constriction. So there's an invagination being formed. And if you increase this bending force, well, then you get something that's more robust and still more or less complex. But we're exploring to which extent this works, because it works for this morphology. It doesn't necessarily work in general, just to some extent. Okay. So we also found that the fact that cells divide by itself has an effect on morphological noise. So here what we have is just for you to understand, because we found that in the ensemble, but it's not so easy to see. So here's an example where we have this yellow territory. We have 6 cells expressing a gene, and this gene is going to affect, for example, cell contraction or cell division, some cell behavior who is going to produce a change in morphology. Okay, but at the moment there's those yellow cells, they are just dividing. So what happens is that because the direction of division is random, this territory starts from having a quite regular shape, but over time acquires this kind of messy, rough, or sinuous shape. Okay. So what happens then is that when those cells affect cell contraction, they actually lead to a morphological noise. So in a way, the noise in the boundary between territories translates into noise at the level of morphology. So that's what we show here for morphologies in the ensemble. Each point will be a morphology. We plot robustness against how rough is the shape of territories. So for example, here you see that this yellow territory is rough, but not too much. It still has a more or less straight line, while this one here is really rough. It's just complete mess. Okay, so you see that there's a correlation between this roughness at the level of gene expression in 2D, 2.5, with morphological noise. Okay, not very surprising, perhaps. However, we found if we actually polarize the division of division, so you make that cells don't divide in random directions, but just always in the same direction, we still have the same effect. Okay, so it's not exactly the direction of cell division, but just the fact that cells, when they divide, they need some space, and this space is found somehow in a random direction. Well, I could explain you better why, but it's tricky and I don't have that much time, so I will skip it. Okay, so then because of the contour of the territories have an effect on morphology, then it happens that the shape and size of territories has also an effect on morphological noise. So essentially, when there is more contour per area, there's going to be more noise. Essentially here, this territory is actually larger. So when it grows, it gets some noise in the contour, but the amount of noise compared to its area is not so large. It's just pure geometric thing. It's not very complicated or something, okay? So the more small the territory is and the more it grows, the more noise is going to induce. And the same happens for sharpness. You know, you have a very narrow territory, that's going to produce a lot of noise. If the territory is a bit more wide, that's not going to produce as much noise, okay? By the way, the same happens for gradients. I don't have the figures, but instead of having a discrete territory, you have a gradient where gradual changes in gene expression, you get exactly the same thing, okay? It's the same problem somehow. Then we get that cell signaling has a strong effect on noise, but the effect is quite complex, or no, not quite complex, but a bit ambiguous. Short-range signaling actually increases morphological noise. So here we have this territory with this funny shape, and these yellow cells, what they do is they promote the secretion of a growth factor. And here what we plot is the concentration of this factor, here, here, and here. And then here we plot the territory induced. So this growth factor actually promotes expression of another gene in the cells that receive this growth factor with enough concentration, okay? So essentially this territory is inducing this one over here. If the signal doesn't diffuse much, what you get is a copy of this noisy shape, but a bit in a larger area of space. So essentially what you're doing is you're increasing noise by copying your noisy shape into a larger area of the embryo, okay? However, at a larger scale, if the growth factor diffuses more, what happens because of pure physics is that the shape gets average. So essentially, diffusion works that essentially you in a given cell actually gets molecules from the neighbors based on whether its concentration is actually larger or smaller than the neighbors. So essentially, diffusion has an averaging effect. So essentially, what you do is you actually lose somehow this shape. Essentially, you lose spatial information, but in this case, it's good because this spatial information is quite noisy. So actually, you induce a territory that is actually more regular. So in a way, you use information, but that's for good. You become more robust. So essentially, a long range signaling actually decreases morphological noise. And then we also have this funny situation where actually, if you have a gene network in which a gene activates itself, either directly or indirectly through a positive loop, and if this gene or this loop, it doesn't involve any extracellular signal, then you actually increase noise. So here what we have is these yellow cells here, they actually express a gene who promotes the secretion of a growth factor, okay? So actually, this gene is gene 1, who's a growth factor, and induces gene 2, who's also a growth factor, and gene 2, it binds to its receptor and so on, and it promotes its own synthesis, okay? So over time, we found this distribution. So the concentration is quite high here and low here. But it's noisy, but not so much. If we do that gene 1 actually diffuses in space, but gene 2 is a transcriptional factor or some gene within the cell who activates itself, then we get a lot of noise. And this happens because this gene cannot actually respond to the averaging effect of diffusion. So essentially, once it gets its kind of loop, it's somehow insensitive to signals. So then when this cell over here in yellow divides, in this case, it's not a big deal because it will change its expression based on the concentration. It talks to each other and it decides that, well, I shouldn't express myself so high. Well, this one, he doesn't hear to its neighbors. So essentially, his expression is autonomous. And when the cell divides, it produces this kind of clone surrounded by cells who are have a different concentration. So this actually produces a lot of noise. Okay. Good. The same happens, well, we also get a lot of noise, and that's from some old work, if we actually activate the stem cell behavior in a large area of the embryo. Okay. For example, here we have those cells who contract, and you actually get some nice imagination. If you actually contract many cells at the same time, you actually get quite a noisy thing. Okay. This is because of just a geometrical reason. I mean, the amount of contraction depends on, well, the contraction of a cell determines how deep the invagination is going to be. So if you have a lot of cells contracting, there is no way to produce just a single invagination. You're going to produce many. And if all the cells are the same, these invaginations will form in random positions. Okay, so in a way, if you want to produce an invagination over a large territory or a large field of cells, you need to partition this field into different territories and different levels of gene expression of something. Otherwise, it's going to be a complete mess. Okay. Good. So essentially what we found is that how much noise we should expect from the normal behavior of the classical or the basic behavior of cells in development and how they interact, okay? And then in a way, we don't explain how we become robust, but in a way, we think that in order to become robust, you have to affect those things that produce noise, okay? And in fact, to the question, so is there some buffering mechanism or not? So results seem to indicate that, yes, to some extent, there should be something special. There's some way to make the development should be robust. Otherwise, if you just have the normal development, as we see in the model, there's too much noise. So how should this buffering mechanism be? We are working on that. So how this development evolved to be more stable, but that's a story for another day. I will just tell you that we are working on that. Some of the results we found is stuff that people have been proposing. So these buffering mechanisms are not necessarily very fancy. Okay, it's just some interactions in development whose function is not so much to make development to proceed to construct the body, but to make this construction more robust. Okay, and we're working on that. So next time I can tell you something more interesting. So this is actually published. Some of the stuff is published. How to become more robust is not published. We're going to publish it probably soon. We have the paper. Just as a final discussion or explanation of what we're trying to do. So essentially, I'm trying to find myself in the ensemble. And by myself, I'm trying to see all the kind of things that we should actually need to produce complex morphologies. So in addition to this ensemble approach, we're also trying an evolutionary approach where we're actually evolving morphologies pretty much in the way Renske explained before, but with embryo maker, okay? And we're also trying ensembles with more genes and different phases and so on and so on. So we have plenty of people working on that. And well, some of those actually. So this is mostly work by Hugo and to some extent Tasio, and to some extent Pascal. Yeah. So that's it. Thanks.